By training, I'm an infectious disease biologist and epidemiologist, and my research group studies how different infectious pathogens, including both animal and plant viruses, evolve and adapt to new hosts. My particular specialty, though, is on using genomic sequence data to track the spread of pathogens. Just like us, viruses have genomes that serve as instructions for all the genes and proteins the virus needs to complete its life cycle. Viral genomes are composed of DNA, or, as in the case of coronaviruses, RNA, and thanks to new technologies, we can now fairly easily sequence viral genomes, revealing the exact sequence of nucleotide bases, the AACs, Ts, and Gs. These genomic sequences are now being used to study the origins, emergence, and spread of SARS-CoV-2, the virus behind the current COVID-19 pandemic. How sequencing the genomes of these viruses has given us new insights into the evolutionary origins and emergence of SARS-CoV-2. Let's begin by looking at the coronavirus genome. The viral genome is about 30,000 nucleotide base pairs long, which is about 100,000 times smaller than our own genomes. Still, the virus genome contains all the genes it needs to replicate itself and live inside our cells. The first 2/3 of the viral genome has instructions, or encodes, for enzymes that copy the virus's RNA genome, allowing the virus to replicate inside our cells and produce viral progeny. The remaining third of the genome encodes for what virologists call structural and accessory proteins that allow the virus to carry out the rest of the viral life cycle, including proteins like the spike domain that the virus uses to enter and bind our cells. Now let's look at the actual genome sequence of some of the coronaviruses in the same viral lineage as SARS-CoV-2. Different viruses are shown here in different rows, and their sequences are aligned such that the columns represent the same genomic position in each virus, so we can easily see how similar viruses are at the molecular level of individual nucleotides. When reduced to this alphabet soup of ACs, Ts, and Gs, these viruses don't seem all that impressive. But by learning how to interpret these genomic sequences, biologists have learned a great deal about the origins of coronaviruses and how SARS-CoV-2 ultimately came to upend our entire world. The key to this story lies in the mutations, the small changes that led to the genetic differences among these sequences, and what these mutations can tell us about the virus's evolution. By looking at the mutations that differ between two viruses, we can get an overall sense of how genetically similar two viruses are at the molecular level. The plot that I'm showing you here shows how similar a SARS-CoV-2 genome, taken from a patient in Wuhan in January 2020, is from other coronaviruses sampled from both humans and animals like bats. Across most of the genome, SARS-CoV-2 is over 95% genetically similar to a sequence sampled from a Chinese horseshoe bat in 2013. Virologists now call this sequence RATG13, a mashup of the acronym for the bat species the virus was sampled from, Rhinolophus affinis, and the year it was sampled. The discovery of these SARS-like coronaviruses in bats back in 2013 perhaps should have sounded more alarm bells. Humanity already had one near brush with death experience with the first coronavirus in the early 2000s. About 8,000 people got infected and almost 800 people died, but fortunately, heroic public health interventions in Hong Kong, Singapore, and Canada prevented SARS from spreading farther. And so the first SARS epidemic was, so to speak, the bullet that whizzed by our ears. The bat RATG13 sequence showed us that there were viruses very closely related to SARS circulating in bat populations, and that these viruses could likely also infect humans. But of course, we all, including scientists and politicians, were distracted by everything else going on in the world at the time. A few years later, more SARS-like coronaviruses were discovered in pangolins, bizarre-looking nocturnal mammals that looked like scaly anteaters. Again, the implications of finding these viruses in an obscure mammal was not immediately clear to most people. However, thanks to the old, pre-scientific doctrine of signatures, the idea that plants and animals look or resemble the medicinal properties they confer, pangolins are heavily traded in wildlife markets based on the assumption that pangolin scales, like mythical dragon scales, could provide strength and protection. The discovery of these viruses in pangolins, like in bats, suggested there was a large reservoir of SARS-like viruses in wildlife, and some of these viruses were likely getting into close contact with people. Looking back from 2020, the genomes of these coronaviruses discovered in bats and pangolins tell an interesting story about the emergence of SARS-CoV-2, although a story that is still being actively debated by scientists. Based on the genome sequences of these viruses and their genetic similarity, we can reconstruct how these viruses are related to one another through a phylogenetic tree. You will learn much more about phylogenetic trees later in this course, but for now we can think of these trees just like any other family tree, or genealogy that shows how individuals are related to one another by descent from shared ancestors. Just as siblings who share the same parent are more closely related to one another than they are to their cousins, or second cousins, viruses that share a recent ancestor will be closer relatives in the phylogenetic tree than other viruses. Because these viruses inherit their entire genome from their viral parents, we can then infer the phylogenetic relationships based on genetic similarity. The viral phylogenies of the SARS-like coronaviruses tells a fascinating story that we can somewhat simplify here by only looking at the lineage that gave rise to SARS-CoV-2. Over the vast majority of the genome, SARS-CoV-2, represented here by just a small number of representative samples from Wuhan in pink, is most closely related to the bat RATG13 virus in green, suggesting that the virus emerged from bats, possibly from a spillover event directly into humans. But in one critically important region of the genome, the spike domain that allows the virus to bind and enter human cells, SARS-CoV-2 is actually more closely related to the pangolin viruses and includes several mutations that are believed to allow the virus to more efficiently infect human cells. Based on these similarities, at least two groups of scientists have argued that pangolins served as an intermediate host in the emergence of SARS-CoV-2. So we are left with conflicting evidence for two plausible origins of SARS-CoV-2, the directly out of bats hypothesis and the intermediate pangolin host hypothesis. One highly plausible theory is that either the human or bat RATG13 resulted from a recombination event that combined different viral genomes together to create a mosaic sequence. Based on this theory, the human SARS-CoV-2 virus could have acquired the majority of its genome from a virus in bats, but the spike domain region from pangolins or another host. What is clear based on the genomic evidence is that SARS-CoV-2 ultimately originated from viruses commonly found in bats, whether pangolins served as an intermediate host or not. Moreover, the mutations in the spike domain that appear to have pangolin origins tell us something else important. SARS-CoV-2 almost certainly did not escape from a lab, nor was intentionally released. While virologists have clones of SARS-like viruses that they study in the lab, none of the known lab strains have the pangolin-like mutations in the spike domain. It was not even appreciated before the emergence of SARS-CoV-2 that a virus bearing these mutations could infect human cells. Combined with the high genetic similarity of the SARS-CoV-2 virus to viruses that naturally circulate in bats, this provides strong and compelling evidence against an accidental or intentional lab release. However, we still have not found the smoking gun. While the bat and human SARS-CoV-2 sequences are closely related, the remaining 5% difference amounts to quite a few mutations across the entire viral genome. And based on the rate at which mutations accumulate in these sequences, it would have likely taken several years for all these mutational changes to occur. I, like most other virologists, suspect that as we sample more coronaviruses from bats and other wildlife, we will find viruses even more closely related to SARS-CoV-2, and these viruses will provide further insights into how SARS-CoV-2 entered the human population. However, thousands of viruses have now been sampled from infected individuals in the human population since the beginning of the epidemic. And thanks again to mutations carried in these viral genomes, we can reconstruct another phylogeny. This one showing how viral samples taken from different infected individuals all over the world are related. Viral phylogenies can therefore tell us the story of how an epidemic spread through a host population. If we know the geographic location of each sampled host, we can use the phylogeny to link cases across locations. For example, if cases in Seattle are clustered together with cases from New York City in the viral phylogeny, we know that the virus must have moved between Seattle and New York City at least once. The key insight here is that we can use the viral phylogeny to link cases in different locations, and thereby reconstruct how the virus spread in the human population after its initial introduction in exquisite detail. This video shows the spread of SARS-CoV-2 around the world as reconstructed from viral phylogenies. The virus began spreading in China in late 2019. While a lot of attention has been focused on the seafood market in Wuhan, where public health officials were first alerted to the virus's spread, it seems as though the virus may have been spreading in the human population weeks or months before the first seafood market cases. As we now know, the virus then spread from Wuhan and China to other countries like South Korea, Vietnam, and Japan. Because of our well-connected global transit network, the virus then spread to Europe and North America, and throughout the rest of the globe. This type of molecular or genomic epidemiology will likely play a very important role in our understanding of how COVID-19 is spreading and evolving in the months and years to come. Beyond just providing historical insights into viral emergence, we can use viral phylogenies, together with epidemiological models, to figure out how the virus is spreading in nearly real time, or at least as fast as we can sequence viruses. This will allow epidemiologists to figure out where new infections are coming from, and whether there might be source populations responsible for disproportionately large numbers of new cases that could be targeted by public health interventions. In the longer term, SARS-CoV-2 will likely also continue to evolve and likely adapt to transmission in humans. Moreover, as immunity builds in the human population, either due to natural immunity or hopefully very soon a vaccine, the virus may also evolve to escape our immune responses. Predicting how the virus will evolve is very challenging, but in my podcast, I explore ideas for how we can go beyond using viral genomes and phylogenetics to reconstruct the past and begin to predict the near future.